Thank you, Wilton, and I'm gonna do. Okay, so I'm a third-year PhD student, and I study how climate change in mountains impacts the nitrogen cycle. And I feel like I always forget to talk about why the nitrogen cycle is an important thing to study, so I'm just gonna insert that in here with this beautiful picture of the mountains. So nitrogen is an essential nutrient for plants, but what do plants use nitrogen for? It's essentially the building blocks of amino acids that make our proteins. And it's also what makes this landscape green. So what makes the sky blue? That's complicated, but what makes what makes plants green? It's nitrogen, and so that's the nitrogen that's in the chlorophyll that enables photosynthesis, and it's the nitrogen in the enzymes that power photosynthesis. So it's important in our proteins as well, and is in nucleic acids, which make our DNA. So it's a really essential nutrient, and without nitrogen, you could not have biological life. And this nitrogen cycle, which you've all seen probably starting in middle school, all of this, every single part, every arrow you see here, every box you see here is impacted both by climate, but both by biotic things. So a lot of this, I call this kind of the recycling system, the nutritional recycling system of an ecosystem. So as you have these green leaves fall to the ground, these inputs, there's a lot of microbes. For every one of these arrows, there's a different type of microbe that breaks down that nitrogen into smaller and smaller parts and recycles it to a form that's available for plant uptake. And as things get warmer, these things happen faster. Microbial metabolism is faster. But another thing that affects this whole cycle is the biology. And so it's both the microbes that are doing these processes, but also the plants that are there contributing to this process. So what kind of leaves are we getting put into the system? What's the quality of those leaves? How much nitrogen is in those leaves? And climate change is going to affect both parts of this, both the biology and of course, the abiotic environment in which nitrogen is cycling through a system. And we know a lot about this direct pathway. There are plenty of studies that show what happens when you heat an ecosystem up, what happens to the nitrogen cycle. And examples of that, some that I use in my dissertation is like an open top warming chamber. So think like a mini greenhouse without a lid on it, and it passively warms up the ecosystem. This is from Hubbard Brook, where they've installed heating tables in the soil. And so that's heated up the soils in these plots. So these plots where there's no snow, they're hotter because they have these heating tables. Or we have these heat lamps. So they're just kind of pumping infrared heat actively and heating up these plots at the Rocky Mountain Biological Lab in Colorado. And what we find is that when you heat things up, microbes metabolize things faster, you get more nitrogen cycling faster through these systems. But the piece that we don't know really is how these biotic changes that will happen with climate change, how that will influence nutrient availability. So these biotic changes, it's getting warmer in places, it's getting drier in places, that's going to impact the plants, and the plant community is going to shift in response to these abiotic changes, and these changes in the plant community will in turn affect nutrient availability as kind of the physiology of these plants changes in response to climate. And we know a lot less about that. We have all these theories about how different parts of the plants impact different parts of the nitrogen cycle. So fixation, where you have plants working together with some microbes in their roots to pull nitrogen out of the atmosphere and into a form that's available for plant uptake. We know a little bit about how changes to plant nutrition can change this process, how much they're willing to pay microbes essentially for nutrients. We know a little bit about how climate change affects and these different physiological changes in plants affects nitrogen losses and denitrification, so that's this side of the cycle. And we know a little bit about how it's plants and these inputs and the changes to the chemistry of their leaves is impacting this recycling process, this nitrification, where these leaves are broken down into nutritional forms of nitrogen that can then be taken up again by the plants. And so, I mean, you can think intuitively about how climate change will impact plant communities. So if you imagine each of these dots is a plant species in your heads, so you can think that climate change, if it gets warmer or drier, essentially in an ecosystem, you might lose a plant species locally. It might go locally extinct. That plant can't tolerate this new climate. So that plant community will have different plant inputs to the soil, to the nitrogen cycling system. You could also have novel plants that are moving their distribution to track this new climate. So the classic example in mountains is that plants might move upslope to kind of track their thermal niches. And you could also just have changes in abundances. There are going to be some plants that are better adapted to taking advantage of this new climate than others. And so they can change in their abundances, but they can also change in their biomass. So all of these changes affect what kind of leaves are falling into the soil system. And these are the plant inputs that are feeding this microbial recycling system. And so we've developed a series of experiments to test this. How will these shifts in plant communities impact the nitrogen cycle, or impact any type of ecosystem function, really? And that experiment is a removal experiment. So humans just go through and kind of, sometimes randomly, sometimes because they're interested in the effect of a specific plant species, but they'll remove a plant species from an ecosystem and see how the ecosystem then responds to the loss of that biodiversity, and how that impacts both function and how it affects the structure of the rest of the community sitting around, and that absence. And so when I first started my dissertation, I said, I'll just do a meta-analysis, because surely people have done this before, all of these removal experiments, and they've measured nitrogen in response to the removal. It turns out there's not enough data there to justify that. So I decided to do my own kind of pseudo experimental meta-analysis, if you will. So in Colorado, at the Rocky Mountain Biological Lab, I took advantage of 4 removal experiments that are already going on across an elevation gradient. So in Colorado, you have some sites that are at this low elevation, Almont, so it's up about 7,000, 8,000 feet, all the way up to Cinnamon Mountain at about 3,500 meters. And a couple of sites that are relatively close to each other in this kind of mid-elevation gradient. The other thing is that this elevation gradient is a moisture gradient. So I'm not going to say it gets dry to wet, but it gets dry to less dry. So Colorado is very moisture limited. That's the main thing that kind of limits function in this ecosystem. And we definitely have from this like kind of sagebrush steppe environment to a more really moisture abundant, more diverse alpine meadow at the highest elevation site. And we also have differences in the type of plant that's removed. So at some sites we remove this leafy green sunflower form here, sometimes a grass, sometimes an invasive plant, this Linaria vulgaris. And some of these sites also have this random biomass removal. So the criticism of removal experiments is that if you remove a plant, that's going to cause some kind of disturbance, and so the effect you see of that removal experiment might just be because you're removing some kind of biomass. Some of these experiments have a random biomass removal control for that. So the take-home message here is that all of these experiments have a really different ecosystem. You're moving a really different plant. So you should think that nitrogen mineralization at each of these sites will kind of be impacted differently by these removals. To test that, Carrie helped me with this. We took soils from all of these studies and we put them in these incubation jars. We brought them up to ideal moisture. So this is the potential part here. We're kind of testing the overall capacity of these soils, not like what's happening in situ. You might expect if we just took soil straight from the field at Almont, because it's drier, it's going to have lower mineralization rates. So the fact that we brought them up to ideal moisture is important. And what we found was actually really surprising. So these are the total mineralization rates that we found at each of these 4 different sites. The red bar is the control, and the blue are the removal. And we found that, first of all, site doesn't matter. So despite the fact that the climate at each of these sites is really different, and despite the fact that a different type of plant was removed in each of these experiments, it doesn't matter where you are, what plant was removed, nitrogen mineralization declines with that removal. And maybe even more striking, the thing that pops out to me in this graph is that the variation decreases. So the height of these bars in the removal plots really goes down significantly when you remove a plant species, regardless of what you remove. And so there are a couple of mechanisms that was proposed by Sandra Diaz and others to think about what might be causing this response. One is that it could just be a response to the loss of the specific species and the job that species does. And when that's gone from ecosystem, you're seeing that effect of the loss of that role. It could be that it's the effect of how the other community kind of reshuffles, the other community members reshuffle in response to removing that other species. Or it could just be you see an effective disturbance. And so we can kind of use our experimental design to rule out, honestly, all of these, but here's why. First of all, because we don't see a difference of any of these experiments where different species were removed, we can kind of rule out that it's not a species-specific effect because it occurs regardless of the species you remove. This graph is kind of the shape of the plant community. So you take all those different species and you can compress it into 2 dimensions. And so if these shapes overlap, it shows that the community structure really isn't changing with the treatment. So the orange here is the removal, the green's the control. And what we find is that the plant community isn't really different in these removal plots. So it's not this reshuffling of the plant community that's really driving these changes. And we don't really know about the disturbance effect. So when you zoom in to only these 2 plots that have this random biomass control treatment, we find that it's not really different from the control treatment, but also we don't see a removal effect. So it's kind of unclear what's going on here. But I think there's another type of, here she comes. There's another type of disturbance that I think might be going on here, and that's resilience. So this is Almont in a really wet year this summer, and this is what it looked like the year I sampled. And so what we could be seeing is that when you remove a plant species, it really kind of decreases the resilience of the entire ecosystem. And that might be what's going on in a bad year, in a bad drought year, because you've lost some biodiversity and maybe some of the stability that that species provides in the community. You might be losing function as well. And I'll just stop there.
Well, and yeah, we have time for a few questions. We're not like crazy rush, but I just wanted to make sure people could have time to discuss. So thanks.
Yeah.
Great talk.
Thank you.
I was wondering whether you're going to repeat this experiment in future summers to kind of get at the point at the end.
Yeah, so I mean there are a couple of things that people have brought up that we could look at to think about this resilience idea. So I got this idea from a removal experiment that was done in Sweden over 30 years, and they saw that and this was not for nitrogen mineralization, but for just above ground productivity, another important function. They found that if you looked at like biomass and a control and a removal plot at any one year, you didn't really see a difference. But if you looked at it over a long period of time, you saw that in the removal plots, you got really big fluctuations in the biomass. And so they said that kind of resilience was therefore kind of decreased when you removed one of these species. And so I thought that might be what's going on below ground. We can look at that though. We can look at to see if that effect is going on above ground first. We have plant data from these different years, and we can see if plant biomass change from year to year. Another thing that might be going on is just like root immobilization of nitrogen. We clip these plants at the above ground level, and the roots are left below ground, and it's going to take a while for those roots to decompose and release the nitrogen in those roots. So what we might be seeing is just that, well, you remove a plant and you lock up a bunch of that nitrogen below ground. Could be what's going on, but yeah.
I have one.
Yeah.
Like, if you got the plants from different elevations, but then you brought them all to the same like potential, wouldn't it be important to study like if those plants would never have that high potential? Like, was it based on temperature or moisture?
It was moisture, and it was incubated in a lab, but the moisture conditions were conditions. It's kind of like the optimal moisture that 30% moisture. All of those sites reached 30% moisture at some point in the growing season. It just wasn't necessarily that's a temporally dynamic thing. So this is kind of just like saying on a good day, what can you do? Soil microbes, how much nitrogen can you mineralize? Regardless of what the soil was like when we sampled them. That makes sense.
Okay. Well, yeah. One more question.
That's good. It was about Alex Morgan. I'll ask later.
Sorry. Yeah, you guys can find Kenna after. All right. Thank you, Kenna.